All right, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요. And hello all. Welcome and thank you, SynBYSS people, for joining us the 105th seminar, and I call it a USDA Day. I cannot believe it's already the last day of November, and I heard many university very busy, people very busy, just finalizing their semester, final exam, etc. And of course, only one more month is remaining this year. This week, I finished one seminar trip, and only one more seminar tour in West Coast is waiting for me to give 4 more talks this year, and I'm done this year. It has been another crazy year with more than 150 days out of town, 2 years in a row. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Dr. John Erickson. He is a program director at United States Department of Agriculture, National Institute of Food and Agriculture, called USDA-NIFA. Among many, he is interested in biocontainment and biosafety, supporting one of my projects. So thank Dr. John Erickson. Also, thanks so much for your leadership and support to the scientific community by providing your vision and research direction, and also your engagement with the community people. And the virtual podium is all yours now. Thank you.
All right. Thank you for the introduction, TSIC. It's my pleasure. As TSIC mentioned, I am a national program leader. My name is John Erickson with USDA-NIFA. And as he kind of touched on briefly there, I'm leading the physiology and plant biotechnology programs at NIFA. And I'll touch a little bit on that. I'm very excited. I don't want to take a lot of time here to hear Dr. González-Grandío's presentation today. It's very representative of the types of things that are of interest to NIFA, as I'll point out here in a second. But I'd like to take a few minutes just to talk about NIFA for those that are less familiar, particularly for maybe early career scientists, and about what we do in some of the programs that we have. So for those that aren't familiar, as he mentioned, we're part of the broader USDA, and we're part of the research, education, and economics area of USDA. And so as you can see in the red star there, that's our NIFA. And so for those that are familiar with the Agricultural Research Service, that's the agency of USDA that does research. And then the National Institute of Food and Agriculture, or NIFA, is the agency that externally funds research through universities and our land grant partners as well as others. And we also have our Economic Research Service and our National Ag Statistics Service that are all part of the REE mission area. NIFA itself is the extramural agency, funding agency. As I mentioned, we have an annual budget of about $1.8 billion, and we award about half of that through capacity funding, which we call. So if you're at a land grant university, you would receive this funding directly through Hatch projects. Supports a lot of faculty salaries and things like that directly. When we also have about half of that goes out through competitive grant funding, and that's what I'll talk mostly about here. Our mission is a little different than, say, NSF or NIH, in that we provide leadership and funding for programs that advance agriculture-related sciences. And so we apply an integrated approach to ensure that these discoveries reach the people that can put them into practice. So we really like those projects that can translate into impacts for growers and farmers in agriculture. And another way that we're a little bit different, NSF, I think, gets one funding line, and then they can decide sort of how they allocate those funds. At NIFA, we get over 80 competitive requests for RFAs in any given year. So we have a number of funding lines. Our biggest one is AFRI, and that's the one I'll focus mostly on here. And this is our Agricultural and Food Research Initiative. About half of our competitive funding goes out through the AFRI program. And some of those, maybe that would be of interest for Eduardo, would include our foundational knowledge of plant products. So this is for things that are focused pathways and physiology and biochemistry of secondary metabolites and things like plant products that may be for industrial or non-food uses, those types of things. One of the programs I lead is the physiology of agricultural plants. We're at a very exciting time now where gene editing and advances in plant transformation really allow us to kind of create plants, impact areas in agricultural plants now more than just model organisms. So that's very exciting for this program. We have our plant breeding program and animal breeding programs that also cover some of this space. One interesting program we have that kind of covers the social implications of these technologies. So for those that maybe have interest or working with those, that program looks at sort of the social and even a little bit of the economic implications of these technologies. And then we have our sustainable agricultural systems, which is part of AFRI, which are larger integrated projects that include research, education, and extension. And then, as TSIC mentioned, one of the grants that he has is in our biotechnology risk assessment grants program. And I'll touch just very briefly on a couple of these, just to highlight a few projects that people might be interested in. As I mentioned, the AFRI program in fiscal year 22, and it's been pretty much the same the last couple of years, about $445 million in total. And like I said, that includes single and multifunction research, education, and extension grants that address key problems of national, regional, and multi-state importance. AFRI-funded projects sustain all components of agriculture, including biotechnology. And these projects also include workforce development and things like that as well. So we have 3 RFAs under AFRI, which is our foundational and applied science RFA, our sustainable agricultural systems RFA, and our education and workforce development RFA, which includes things like postdoctoral fellowships and predoctoral fellowships. So just an example of a project that would have been interested in some of the work that Eduardo has done in the past related to canopy architecture in soybean. We have a project for Dr. Jin Ma at Purdue University that's kind of looking at functionally characterizing genes that relate to branch angle variation and leaflet size in soybean, looking to kind of maximize carbon assimilation that way. Our SAS program, an example of a project-related biotechnology here, is cell-based meat. So this is one of our larger, again, $10 million projects that includes, education component and extension components. And I won't go into everything they're doing here in the interest of time, but yeah, it's one of those interesting projects that's looking at kind of across the spectrum from the technology to the social acceptance to all of those things related to cell-based meat. Our biotechnology risk assessment grants program is another area where biotechnology obviously is the main focus. And the primary thing we're looking at here is anything that will generate information that relates to regulatory needs of these organisms through the EPA, FDA, and USDA regulatory processes. Again, I won't go into the details of what we focus on. Highlight a couple of projects here that relate to this program. This covers animals, plants, and microbes, anything that's been bioengineered. And so we have an example from University of California that's looked at the comparative evaluation of the phenotype, the genome, and animal products derived from offspring of a genome-edited hornless bull. Washington University, a TSIC's project for development of kill switches. We had a couple related to this for biocontainment of genetically engineered microorganisms. And at NC State University, they've implemented the first gene drive in an agricultural pest and are evaluating that as well through this program. So that's an exciting project as well. One of the projects that's kind of been funded through capacity funding, I think some AFRI funding, and also BRAG funding, is a project focused on restoring American chestnuts. They've used a gene from wheat that's brought in some resistance to a blight fungus, and are currently going through the regulatory process to look at being able to start planting this chestnut again, and hopefully restoring chestnut to some of the Northeast. And I wanted to wrap up with an opportunity that is currently open that may be of interest. It's an international opportunity that's entitled Future Proofing Plants, and it involves the DFG in Germany, the BBSRC, UKRI, and then in the US it involves the National Science Foundation and NIFA. And so in this opportunity that's currently open, we're currently at stage 1 here, soliciting expressions of interest. This is going to be led by the BBSRC, but it's going to involve scientists from all of the partner countries. So you have to have applicants in your application that are in Germany, in US, and in England or UK. And so that project is currently open, and one of the themes under that is programmable plants. And so the ability to program plants with predictable and novel characteristics and idea types suited to specific environments that include, synthetic or biotechnological approaches to accelerate development and climate adaptation or climate resilience. So that may be of interest to some of this audience as well. And so with that, I just wanted to wrap up, as our USDA non-discrimination statement that USDA does not discriminate and is an equal opportunity provider, employer, and lender. And I don't know if I can see them, but I'm happy to stick around and take questions later if people have any questions about any of these opportunities, or I don't know if they can pop them in the chat or how you want to handle that, Tae-Sik, but that's what I want to do. And I'm looking forward to Eduardo's presentation.
So thank you. That's amazing and very useful information. Q&A will be done after Eduardo talk basically. So if you, I'll chat. I'll put some in the chat later. So that's absolutely brilliant. And then especially I like the international collaboration part, because I strongly believe current climate crisis cannot solve by one country. That is the global problem. So USDA starting those type of international collaboration is really, really fantastic. In that way, we have the more engagement between the different countries to solve the one global problem together. So absolutely amazing. Thank you. Okay. Now, the main speaker of today with a longer introduction, Dr. Eduardo González-Grandío received a biochemistry degree at the University Autónoma de Madrid, Spain, and then pursued his PhD at the Spanish National Center for Biotechnology in the laboratory of Pilar Cubas, studying the gene regulatory networks that control branching in Arabidopsis. In 2016, he moved to UC Berkeley to study chromatin remodeling in response to light in the laboratory of Peter Quill. Afterwards, he did a second postdoc at UC Berkeley, developing a DNA delivery method based on carbon nanotubes at the laboratory of Makita Laundry. He moved back to Spain thanks to a Marie Curie fellowship. Then he obtained a Ramon Y. Cajal contract from the Spanish government, and now he's starting his independent line of research on plant synthetic biology. You know, based on what John mentioned, I guess this is a good match. I mean, between these two speakers, you know, they're interested in both, I believe, the plant science or engineering. So I'm super excited to hear his talk. So Eduardo, thanks so much for your time today, and please take it away. And it's all yours now. Thank you.
Can you see my presentation
Yes.
now? Okay. So thank you for the introduction and for the opportunity of showing my research and my current project. So as you mentioned, I wanted to talk briefly about my background and where I come from. And as you mentioned, I did my PhD studying the genetic control of shoot branching in Arabidopsis thaliana, that is a model plant. And there I studied one gene that's branch one. That's a transcription factor that really regulates branching. And I described how this transcription factor eventually controls ABA, abscisic acid synthesis, in axillary buds to regulate branching. During this time, I worked briefly in tomato, also in branching in tomato, because the idea was to translate all the knowledge we generated in Arabidopsis to a crop or to a plant of agricultural interest and apply our knowledge. And I'll talk briefly how this has developed now. Then, yeah, I moved to UC Berkeley with Peter Quayle. I studied chromatin remodeling. In my PhD, I studied mostly transcriptional regulation, and here I wanted to learn about another layer of gene regulation of chromatin. And then I studied how chromatin changes in response to light. And then for a second postdoc, I work in a much more applied biotechnological project. And because always gene editing has been part of the tools I was using in my previous projects, and something that I think is really of interest, and in the next years will be a very important technology in agricultural development. So the idea was to generate a new technique to deliver these gene editing reagents, CRISPR-Cas, for example, by carbon nanotubes. So this technique is still in development, but I think it holds great promise, and hopefully in the next few years we can see like results of this technology. Then I moved back to Spain after 6 years in California with these 2 fellowships, and I started developing, further developing the project in tomato, regulating the control of shoot branching. I initially was going to talk about this project in this talk, but we are collaborating with a company, and they advised me not to share our very exciting results, because they always say that scientists, we always want to share our results, but sometimes it's not the best, especially when intellectual property is at risk. So I'm going to talk about this other project, is the generation of plant biofactories for the sustainable production of mosquito repellents and other high-value compounds. So this project is in its very early stages of development. I recently got more funding, so hopefully I can start hiring people and accelerate the project. But I want to show the initial results I have, and also I'd like to obtain some feedback on alternative approaches to do what I'm going to do, or questions or any other feedback in general is very welcome. So why use plants as biofactories? Well, of course, as you know, plants are already very good natural biofactories. They use sunlight, water, and CO2, and they produce oxygen and glucose. So they are sustainable in a way, and they are very efficient. It's, in fact, like 80% of the biomass on Earth comes from plants. So they are very, very efficient, and they use just sunlight and water. So the idea is to try to harness these natural biofactories to produce more things other than glucose. And these are like secondary metabolites. As you can see here in this huge map, at the top in the red square, that's glucose, and glucose, in addition to the energy for the plants, can become very diverse molecules. Many different plant species, even in one plant species, in different organs, this primary metabolism can become secondary metabolites that have many functions. We use some of these molecules as medicine, in fragrance, for taste, for many uses. So the idea of the project is to try to improve, if we can, these natural biofactories into engineered biofactories to produce things that maybe are not produced in a high amount in plants right now, or things that are not produced in plants but could be introduced to be produced in plants. So plants as biofactories, they have several advantages. I mean, the first I think is scalability, because they do not require sterile conditions as other bioreactors like yeast or bacteria. They have compartmentalization at the subcellular and at the organ levels. And this is useful if, for example, some of the metabolites that you want to produce or the intermediates are toxic. So you can confine them in a specific part of the plant and the rest of the plant can grow. And then you can extract it from the part where it's accumulated. And regarding this extraction, in some cases, and you will see later on, the plants can even secrete these metabolites. So it's very easy to retrieve these products. And also they do not harbor human pathogens unlike other methods. Of course, they are not the only advantages. They also have disadvantages, such as they have a long life cycle, unlike bacteria, for example. They are harder to engineer if you want to generate transgenic plants or generate mutants because of this life cycle and the way of the plant cell wall and many other technical barriers. They're harder to engineer. The compartmentalization I mentioned can be a good thing or can be a bad thing. Because, for example, if you want to engineer something in leaves, you have to have a specific, let's say, promoters and gene circuits that do not work in flowers or at the whole plant level. So it's not like in bacteria that you have a single subtype. And right now, one of the disadvantages is generally, it always depends on what you want to produce in the plants, the yield is lower than for bioreactor. But my idea is to try to improve plants or just have an ideal plant that's a good biofactory. So this is an overview of the project. I will go step by step, right? It has different objectives. Of course, I have only advanced in the initial steps. That is the objective one of engineering Nicotiana tabacum, that's the tobacco plant. And I want to have it produce more precursor of terpene molecules. I will talk later about terpenes, but these are a very huge family of secondary metabolites with many uses. So the idea is to have a plant that produces or is able to produce high yields of these molecules. And I will do that by increasing the amount of metabolite precursors and also the trichome density, because in the trichomes, these compounds are produced. Then the idea is, once we have this ideal biofactory, to be able to introduce different metabolic pathways to produce different molecules. In this case, what I want to do is I want to produce one molecule that's PMD, that's an insect repellent, that's produced from citronellal. So I will later explain this metabolic pathway, but the idea is to have different pathways that you can introduce into your biofactory to produce different products. And then at the end, I want to optimize further the production in this biofactory. But let's go step by step. So first, I'll focus on the first objective of engineering the tobacco plants. But why have I chosen tobacco or Nicotiana tabacum? First of all, it has been already bred for production capabilities because of the tobacco companies. So it grows relatively fast for a plant. I mean, it's not like a Arabidopsis, that's a very fast growing plant, but it's not like a tree that takes years to grow. It has a high secondary metabolite production capacity for nicotine, but not only for nicotine, as you will see later on. For many other metabolites, has very high production capabilities. Unlike other plants like a close relative, that's Nicotiana bentamiana, that's a kind of model plant that's used in the lab. but it's used to produce heterologous proteins. It produces them very well, but for metabolite, it doesn't have such a high potential as tobacco. Tobacco can be easily transformed. You can generate transgenic plants with a very simple protocol. Actually, the transgenic protocols were most of them, well, not most of them, but many of them initially developing in tobacco. And at the same time, because generating a transgenic line takes from 6 months to 1 year until you get an homozygote plant, for example, it also allows you to perform transient expression assays. This is done with a bacteria, that's Agrobacterium, where that you can transform. You can insert a plasmid with a gene that you want to express in the plant. Then you can after you transform the Agrobacterium, you can infiltrate it in the leaves of Nicotiana tabacum. And just after 3 days, you can see expression of your protein. In the right, you can see my plants that I have at the greenhouse expressing a red colorant, betalain, just 3 days after infiltration. So in this way, you can really test very different genetic circuits or optimize promoters, many different things that then you can use to generate a stable transgenic lines. But otherwise, if you would test all these combinations doing transgenics, it would take a very long time. Also, I chose tobacco because in the leaves of tobacco, they have glandular trichomes. And that's what I was mentioning, that in some cases, plants can even secrete the metabolites. So these glandular trichomes that you are seeing their microscopic structure, they are several cells long, sorry. And at the tip, they have a glandular cell that secretes products, secondary metabolites. In tobacco, they mostly produce labdaniols and sembranoid molecules. Don't worry about the structure of the molecules. But what I wanted to highlight here is that these compounds can represent up to 10% of the leaf dry weight. That was what I was mentioning, that it has a very high production potential. And these compounds are synthesized at the tip of the trichomes. You can see in the middle, the reporter lines using the promoter of the enzymes that catalyze these 2 pathways, and fused to a reporter, to gas that gives a blue staining. And you can see that they are specifically expressed in the leaves of the trichomes. So imagine if you want to produce one, and here you can see that many other genes that are exclusively expressed at the tip of the trichomes. So this is very good because not only it allows the secretion, allows easy retrieval of the secondary metabolites, but imagine if you are producing something that could be toxic for the plant. If you produce it in the whole plant, the plant would grow less or even die, while you could express it in the trichomes and just have it very, very confined there. So I wanted to talk a bit more about these compounds, that these are diterpenoids. And what I want to produce is a biofactory that is able to produce many more of the terpenoids, that these molecules. So these terpenoids are derived from 2 5-carbon molecules that are isoprenoids. Don't worry about the names, IPP, DMA, PP. And the idea is that not only diterpenes, but also tetraterpenes, monoterpenes, all these molecules are derived from these building blocks. So the first goal is to try to have these biofactories that have higher levels of these precursors, these 2 isoprenoid precursors. So as I mentioned, the tobacco produces high amounts of diterpenes. So the idea is if you block this metabolic pathway, you would increase the amount of precursors that then could be used for production of many other different molecules. So the variety of tobacco that I'm using is K326. It already naturally does not produce labdanoids because it has a mutation in the CPS2 gene. So the idea would be just to block the CBTS pathway to have increased precursor level. And then I'm giving just some examples of the molecules that could be produced by starting from these building blocks. In addition to doing the metabolic engineering of cutting this metabolic pathway, I want to increase the amount of trichomes in the leaves of these biofactories. So there are several genes. I'm just putting one example of a cyclin B gene that when you knock out the 2 copies of this gene, you can see that these plants have an increased number of glandular trichomes. And this increased number of trichomes results in increased amount of ditropins. So these are the two modifications I want to produce: increased precursors and trichome density. And how I'm doing it is I'm using CRISPR-Cas12 system, because Nicotiana tabacum is a tetraploid plant, so it has several copies of some of the genes. So Cas12 can very easily knock out or mutate several genes at once. So I'm using, and I'm showing you later why. So I'm using one variant of Cas12 that has a mutation that makes it thermotolerant. So this is very useful in plants because when you're growing human cells, you're growing them at 37 degrees. So all the systems are more optimal at this temperature, but when you're growing plants, you cannot grow them at 37. You're growing them at lower temperatures. And this mutation makes this Cas12 very efficient at these low temperatures. It also is intronized, so it has increased expression levels. And then I developed a system in which you can express several guide RNAs for the Cas12 to target multiple genes. I won't go into the details here, but I'm going to show you some of the first results of these vectors. So as I said, we can test transiently by algorithm filtration all these different constructs, for example, and see which are efficient to mutate the genes that we want to mutate. In our case, here I'm talking about cyclin B2. There are 2 copies. I call them CyC1, CyC2, and the CBTS, the enzyme that produces ditropins. There are also two copies of this gene. So I targeted these 4 genes using different combinations. I didn't mention it, but I changed the promoter to express the guide RNAs. There's a short version or a long version, just to increase the expression of the guide RNAs to see if it improves editing efficiency. And also I tested the wild type Cas12 versus the intronized and thermotolerant variety. And here you're seeing the indels, the percentage of indels after 2 weeks after algorithm filtration in the 4 genes. And what I chose at the end is the long U6 promoter to express the guide RNAs and the intronized thermotolerant Cas12 that gave the best results. Then, after doing this transient assay and testing of the best variety, and also I test different guide RNAs, I obtained 20 independent transgenic lines, and I genotyped them. And at first, I was very excited because I found 6 lines that had indels up to, if you can see, even like one plant had all the 4 copies, they had indels. But then when I analyzed a bit more deeply the sequences, I found that all these indels were not disrupting the coding sequence of the genes. In some cases, I could find like knockouts for these genes, but in some others, the deletions caused by Cas12 did not disrupt the reading frame. Here you can see 2 examples. If you get a 3 base pair deletion, you're not disrupting the open reading frame, so the protein is, you're removing one amino acid, but the protein is still intact. So I'm still trying to generate the quadruple homozygous mutant because you have to knock out both copies of the cyclin and the CBTS genes. So I'm working on that, like waiting 1 generation. But in these plants, I have already knocked out the 2 copies of the enzyme and performing the metabolic profiling just to make sure that they are not producing this sembranoid, this ditropin. And hopefully I can measure also the levels of the precursor molecules to see if they have increased level. So in the future, once I have this plant, I would like to test the production capabilities by transiently expressing different enzymes and measuring the production of secondary metabolites. Then also I want to increase the biomass production of these plants by delaying flowering. So in annual plants, they have 2 stages of development: vegetative development, where all the energy and resources are used in generated leaves, so the plant grows, and then reproductive development, when then all the energy is focused on flowers, generating seeds and and reproduction. So if you keep a plant in permanent vegetative development by inhibiting flowering, here you can see an example of 2 tobacco varieties. They can grow, I mean, not infinitely, but for very, very long periods and produce much higher amounts of biomass. So how can we inhibit flowering? And flowering is controlled by photoperiod. So usually plants, some of them, they flower in long days or in short days. So this photoperiod is sensed by the leaves of the plants. And I'm not going to get into the mechanism, but this results in the production of a florigen that was called in the beginning. And this is a molecule that travels from the leaves to the shoot apical meristem, and it triggers flowering. In Nicotiana, this florigen has been identified as the gene flowering time 5 in Nicotiana tabacum. And here you can see an example of a plant where you reduce the levels of FT5, so it delays flowering and has increased biomass. It has a triple of the leaf number, and the flowering is delayed by 60 days. And what happens if you knock out this gene is actually what's happening in this Maryland mammoth cultivar of tobacco is these plants will never flower in long days. So the idea is, okay, these plants have very high biomass, but if you want to work with them, they don't flower, it's very hard. And I mean, because you don't get seeds. So in the case, remember the plant I have that is heterozygous, I want to get an homozygote to advance 1 generation, it's impossible. So what people do with these mutants is what they graft a wild-type plant in the bottom. So this wild-type plant is able to produce the florigen molecule, or FT5, and induces flower. But this is not feasible if you want to have this biofactory, having to do grafting whenever you want to have flowering. So how can we induce flowering? So I try to express FT5 in the leaves by agroinfiltration, as I mentioned, but the levels were not enough to induce flowering. So other people have thought of this before. How can we induce flowering? And they have developed systems in which instead of, I mean, where they infiltrate agrobacterium, but instead of having a vector that expresses a protein just transiently, they express a vector that generates a plant virus. And this plant virus can express the flowering time 5 or florigen molecule for longer periods of time. So here you can see an example with potato virus X, where they express FT with this virus, and they can induce flowering. So I'm adapting this system. I'm using a different viral vector, is tobacco retrovirus, because this vector produces higher levels of protein. So the idea is to try to improve this system. So I do not have results for flowering induction yet. I did a brief test just expressing YFP, and I can see that this vector produces prolonged expression. Here you can see 3, 6, 8, and even 14 days after infiltration. You can see still expression of this reporter. So the idea is what I'm testing right now is expressing FT5 with this vector and trying to induce flower. So as you can see, this is only, I mean, like very initial steps of the project. And the other part, I mean, this is going to be at least a 5-year project, probably more. So, but I wanted to give you a brief overview of what I plan to do and how I plan to do. And if you have any suggestion or alternative approach or any feedback, just please let me know. I'll put my contact again at the end, so you can email me or contact me. So what I want to produce is an insect repellent in these optimized plant biofactories. So why an insect repellent? Well, insects, mosquitoes are vectors for many diseases. Well, not many, but for diseases that are quite severe, like malaria, dengue. And preventing mosquito bites can prevent the spread of these diseases. But right now, the available mosquito repellents that have been approved by the CDC and the European Agency, they are all synthesized chemically, except for one. And well, right now it's synthesized chemically. So that's the idea, is to produce this molecule in the plants. But let's talk first about this mosquito repellent, this PMD. And it's very, very efficient. It's as efficient as DET. That's the gold standard. Here you can see the results of the reduction in biting is up to 100%. So this is very, very efficient. And I was saying that this is a natural molecule, but that's produced chemically right now. And I want to explain more in detail how it's produced actually. So what people do is they extract the essential oil of lemon eucalyptus or other plants by hydrodistillation. And this essential oil is very rich in citronellal, but very poor in PMD, just only 1%. And citronellal, although it has some repellent activity, it's very low because it evaporates very quickly and actually is not recommended, for example, by the CDC as a useful measure to prevent mosquito bites. But if you treat citronellal with acids, with sulfuric acid, for example, its structure changes to PMD. And this is what they sell as oil of lemon eucalyptus. That's very PMD rich and it's a very efficient mosquito repellent. So the name might be misleading because it looks oil of lemon eucalyptus. It's not lemon eucalyptus essential oil, but it's synthesized chemically. There are some fungi that can biotransform citronellal into PMD. I'll talk briefly later about that. But right now it's done by chemical synthesis, PMD production. So first, what I want to do is to introduce this citronellal synthesis pathway into the biofactories. But this pathway of synthesis is unknown. It's known that citronellal is synthesized from geranyl diphosphate. That is one of the monoterpene, the main monoterpene precursor, by two probable steps, but the actual pathway is unknown. So the idea is to identify this citronellal synthesis pathway in lemon eucalyptus. So lemon eucalyptus, as I said, has essential oil that's very, very rich in citronellal. From 60 up to 80% of this essential oil is citronellal. So I want to identify it in this plant. So the genome sequence is available of Corymbia sitodora, that's lemon eucalyptus, but the genome that's available is from a subspecies, that's variegata, that does not produce citronellal. It's used for timber production, so it's interesting, but I do not have the genome of the plant that produces citronellal. But I want to find them. So the idea is to use varieties of lemon eucalyptus that produce citronellal and compare them to this other variety that does not produce. And also there are other close relatives, other species of eucalyptus that might or might not produce citronellal. And I want to perform a comparative analysis of these species, trying to identify the genes responsible for citronellal synthesis. So I obtained seeds from Australia because these species are original from Australia, from different locations from the Australia Tree Seed Center. The northernmost ones, they produce citronellal, and the southernmost, in theory, they do not produce, but they have not been characterized. What I'm doing right now is characterizing all these varieties to try to identify these two, the producing and the non-producing ones. So I do this in collaboration with another group here in Madrid, and I collected the leaves, I dried them, I do the hydrodistillation, and I'm analyzing their metabolic profile. So far, I have identified several plants of Corymbia sitodora, of lemon eucalyptus, that produce very, very high amounts of citronellal, almost 90%, and some other varieties that do not produce them. So I'm finishing these experiments because I have up to 35 different plants and varieties, but right now I already have two varieties that could be used for this comparative study. So this is the future steps that I like to pursue, to have a high citronellal variety, no citronellal, to perform RNA sequencing of the leaf RNA, perform de novo transcriptome assembly, because these varieties, they might have additional genes or mutations. I'm going to use the genome that's available, but also taking in account that there might be new genes or duplications or mutations that enable these plants to produce citronellal. And I have some candidate genes according to their function, like dehydrogenase. I won't get into the details. But once I narrow down these genes that could be differentially expressed between these two varieties, for example, I will test them by ion infiltration by transient the expression assays in Nicotiana plant. Then the future step is trying to identify, as I said, the enzymes that convert or biotransform citronellal into PMD, because some fungi have been described to perform this reaction. Actually, there was a group that isolated fungi in the peel of citrus plants in the Amazonas, and these there's kind of like close relatives to lemon, let's say. So these molecules, for example, citronellal, has antifungal activity. It's also used in fragrances for flavor, but also it's antimicrobial, antifungal, has many properties. It's a very interesting molecule. So that's part also of the interest of producing it in biofactories. But in this case, citronellal has biofungicidal activity. So these fungi are able, some of the fungi that are able to grow in these plants is because they are able to degrade the citronellal. And one of these fungi, when it degrades citronellal, it degrades it into PMD. So that's very interesting for us because it's a natural way of converting citronellal into PMD that doesn't involve chemical synthesis that can generate chemical waste. So they isolated one strain of fungi that they identified later as Penicillium bacilli that has a very high conversion rate of citronellal to PMD, up to 85%. The only problem is that this strain is no longer available. So what I'm going to do is order the repositories of fungal strains. I'm going to order several of these and test their PMD production capabilities by just growing them in control or with citronellal. And once I have identified a strain that's able to perform this conversion, I'm going to do again comparative transcriptomics because I expect that these enzymes that degrade citronellal are induced when in presence of this compound, because it has been described for many fungi that when they are in the presence of a toxic compound, they express higher amounts of enzymes to degrade them. And a final step, I don't know how long from now, what I like to do is to optimize the PMD producing plants, because the idea is, okay, I have biofactories, I can introduce the citronellal synthesis pathway, hopefully I can also introduce the citronellal PMD conversion pathway, but there are other ways to further improve the synthesis of PMD or other interesting compounds for which their metabolic pathway is known and could be inserted into the biofactories. I'm going to do this in collaboration with a company that's called Trichofarming. And what they do is they have identified molecules that promote the trichome development and trichome metabolic production. So this is of interest for them for many other reasons, but I contacted them and they were very interested in the project. Of course, they want to see a first proof of concept in my biofactory, but then they are very eager to collaborate and trying to further improve the production capabilities of this biofactory. And finally, I'd like to thank, my only student right now. Hopefully more people will join my lab. I have acquired funding right now. I have open positions. If you are interested or know someone that might be interested in pursuing a PhD or doing a postdoc in this project, please contact me. I have funding available. And then, I'd like to thank the Pilar's group for hosting and our collaborators, Diego Orsay for helping in gene editing, and the biopesticides groups for all their help in metabolite characterization. Because of my expertise, I'm completely new to this and they're helping me so much. And if you have any questions, any feedback live, or you can comment on YouTube or send an email or contact me on Twitter if you like.
Fantastic. Thank you so much for your amazing talk. I really enjoyed. Before I ask any question, I want to ask John whether he has comment or questions.
Not really. It's very exciting work. I guess I did have one kind of comment of maybe outside a little bit of the scope of work, but I'm just a little curious how you get from the trichromes to the product and what's the process for that?
Yeah, so there are protocols for extraction from the leaf surface that, in principle, at least at low scale, look very simple. It's just dipping the leaf into hexane, and then just you have the product if it's soluble in hexane or maybe other organic solvent. The only, issue would be like scalability of this extraction procedure. But hopefully when we get there, we'll see, like we'll do like an techno-economic study or something like that. But first, I would like to have like a proof of concept that this works, it's able to secrete, and it's, at least in lab conditions, it's easy to retrieve.
And then whether there'd be like any chemical waste associated with the extraction process relative to maybe synthesizing it, be an interesting comparison.
Yeah, that's the thing, because with hexane, if we are trying to avoid using sulfuric acid for the production of PND, but we are using an organic solvent for extraction, you have to wait what's more sustainable.
Right. Excellent
really.
presentation. Very excited to see the results.
Me too.
Thank you. So one thing I want to ask is, I strongly believe plant engineering also, I would call it plant synthetic biology, lots of research necessary, but in some sense, unfortunately, not many people working on that. The reason I believe is, even myself, plants grow very slow. At the same time, engineering tool available for model organism, but non-model organism, we still develop a lot of tools to engineer. So you are expert in plant synthetic biology, and my question is to encourage other colleagues, especially younger colleagues like student, graduate student, and potentially another postdoc, to jump into this challenging field. What could be your kind of advice, or based on your experience? Because even me, I'm in senior, not getting becoming senior researcher, but still I want to work on plants, but still I rely on my collaborator rather than I'm jumping into the field. So could you kind of share your advice or insight? What is the best way to approach the plant synthetic biology or research?
Yeah, I have to say, that tool development in plant synthetic biology is always lagging behind.
Mm-hmm.
So mainly, we are mostly like profiting from other fields and then applying all those tools and technologies to plants. But right now there are several groups that are starting to develop things that are plant specific. So that's been very useful, like promoters, gene circuits, all these things that when you translate something from other organisms, it might work in plants, but it might not be the best. So my advice would be try to see what's been useful in other organisms and try to develop it in plants, but not coming from these other organisms. Just, using plants to develop the tools, not just using them from other places. Because actually these tools are proving to be very, very useful. It's like, for example, when you mention life cycle, I want to shorten the life cycle of these plants, at least when I work with them. So that's why I'm trying to develop these viral induced flowering tools that having already developed, but I'm trying to improve them to have shorter cycles in generating plants. So maybe that can help develop other technologies that are not developed right now because of these long cycles that take a long time to develop and that kind of things. So that's why I'm trying to develop tools and applying them at the same time, but developing them specific for plants. Like
that's fantastic. So I actually this week I visited to give a seminar, but I met multiple plant scientists, and then I was so excited the potential also their research kind of potentially generate in the future, because they are working on the soybean, and the soybean also, as interact with the bacteria that fix nitrogen. In that way, they said the reduction of the chemical nitrogen fertilizer is super, a lot. In that way, we don't necessarily need to worry about chemical fertilizer kind of destroy, ecosystem, etc. At the same time, potentially increase the crop yield in a developing country and so on. So I believe agriculture is very important. Still, there are lots of, engineering, should be done, but a little bit, lagging behind. But I thank you. Working on plant science, plant synthetic biology. And I want to ask John, do you have any similar wisdom or, the comment on that, my same question? Because you are expert in that area, so you already seen many great scientists in the world. You have the better insight, than any of us, I guess.
Yeah, I think one of the things I think that's interesting about plants is, there's a tremendous amount of diversity there. And so I think there's opportunities for discovery, when you're working with a human genome, it's a human genome, right? It's a single genome, where a lot of plants have multiple copies of genomes. And most of our agricultural crops, or a lot of them, are polyploids and so there's additional challenges, but also opportunities for learning and discovery. And so I think from a research perspective and advancing our understanding that there's things you can do in plants maybe that you can't do as easily in other systems. That's some strengths, I guess. And I think when you talk about the scalability of things, I think it's maybe easier in some ways to scale agricultural plants economically as opposed to large, that's one of the challenges they're facing with cellular meats, right? Is how do we scale this to a process that matches what we already do for agriculture, right? And so that's, I think the scalabilities are often maybe easier in plant systems for some things anyway. So, so there are some advantages. I think, it's just the sheer numbers and amount of funding probably that contribute to some of the challenges we face on the plant side. Our budget is much smaller than NIH or even NSF, right? So that's one of the challenges we face.
That's true. But the one thing I want to tell everybody is the recent years, many universities try to hire plants, engineer or plant scientists. So I've seen many young people even actually speak for this seminar series, including Jennifer Stanford and Caltech, the Gold State Caltech, and also some other senior like June Medford and so on, etc. But they are doing fantastic job. So I hope, Eduardo, of course, Eduardo, working on this important field. So I hope to see more synthetic biologists, working on this exciting field, and then hopefully we have the more younger, generation interested in plants, even though that, you need to wait a lot of a month to see the result. That's part of the difficulty, and that's part of the challenge to me. Because I'm probably impatient to wait to see the data. So
well, that's assuming you can even transform them at all, right? That's the challenge of a lot of agricultural plants as well. And so that, as Eduardo mentioned, tobacco is one that's relatively easy to transform, and there's some others, but it's also a number that are very difficult to transform. And so that's one of the challenges faced by plant scientists as well in this field.
Yes, exactly.
Yeah,
so go ahead.
One of the questions I usually get with this project is like, why go all the way, and can you just put the PMD transforming enzyme into the lemon eucalyptus and you have an eucalyptus that produces the repellent? It's like, I wish I could, but there's no transformation protocol available. Also, working with a tree takes many years. So that's why transferring it to a fast-growing plant that's easy to transform I think it's ideal. But that's this kind of technical limitations is what in plants sometimes makes it impossible to do.
I see. So that is so. Also, because of the people see GMO as bad thing. So also even engineered plants, crops, not that much favorable thing for the multiple aspect, including regulatory and public perception. So we probably overcome those kind of issue as well, I guess. So but still, you know, we need to feed entire population. You know, that is, many people actually even, not that much have the food. I mean, in another side of the globe. So I wish, we could solve this, food security or inequality problem in near future. In that way, everyone not hungry and then enjoy the good food. I mean, that's one thing I want to really passionate about, and hopefully someday, we have that, better world. I mean, okay, so now I see 11:05 already, so I probably need to close today. Okay, so thank you all for joining and staying today, even though I believe many people are very busy. That's what I heard because of the final exam and so on. So we'll meet again next week on December 7th, Thursday, the same time, the same Zoom link. We'll have Professor Ting Nu at UIUC and Dr. Carlota Ronda at UC Berkeley. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. And thank you. And I stop recording, and happy December. Just 1 second.